So the last lecture we were talking about the use of very traditional methods in colony gel electrophoresis based methods of estimating the microbial diversity in environmental samples. Okay. But those methods are really no longer in use unless you are looking for very special occasions. Okay, what are the special occasions? You still use colony approach, cloning approach. You can still use cloning approach when you are looking for rare bacteria. So one where you feel that the database is not available yet, okay, or where the database is not good. So that would uh, then you will need to make your own library of transformants, E. coli transformants carrying the gene that you want from the environment. Okay, because if you were to do the NGS method, it won't work that well because the database will not have the the sequence record that you are getting. Okay, so for specific occasions, as I've also mentioned earlier, if you're doing comparison of sites, qualitative comparison of sites, where you target specific genus or specific family, you can still use DGG or TGG. Can also use cloning. Okay, my student in the past has used cloning to look at the diversity of archaea bacteria in our waters. Okay, so because the database for archaebacterium might not be so strong, so complete, so it's good to have your own longer 16S database. Okay, then yeah, so though you can still use cloning TGG, DGG, and SSCP, but you have to be very clear about what it can do and what it cannot do. Okay, and also at that time NGS was very expensive, 4-5,000 per sample. Now it has gone down to about 400 to 600 ringgit per sample. So the price difference, the price now makes it the price now is so low that it doesn't make sense if you use other methods unless you really need to. Okay, so we are looking at the NGS. In the beginning they use the term NGS, which stands for next generation sequencing. Okay, mainly it is sequencing by synthesis approach, okay, as opposed to the one by Sanger dideoxy termination sequencing by termination. Okay, so but now they prefer the term high throughput sequencing because of the amount of data that you can get from NGS easily in gigabytes. So the amount of data you are getting from NGS is tremendous. That also is one of the reason why every biologist should know how to handle sequencing data, NGS data. Okay, not just bioinformatician, but everyone should learn how to use specific softwares to handle NGS data. The software that, for those who are interested, you can probably look at the following software. Can you see this? The one that I just typed?
Yeah, right there.
Okay, so these are free software. Mother, and also Chime. So these are softwares that are specific to handle NGS data. And the good thing about these two softwares are the tutorials are extensive, and there are a lot of pipelines available. Pipelines are, so they will use a lot the term pipelines. Pipelines are where they give you the flow or processing flow. So they will show you how to process the data, the commands that you need and all that. Even if you're not familiar with programming and scripting, it's not that difficult if you try it out, because it's basically just copy and pasting of the command, or maybe changing a little bit. Usually you only change the directory or the file name. Okay, the rest of the commands are usually the same. Okay. So now they prefer the term high throughput sequencing, and some also use massively parallel sequencing. Later you will see why the term massively parallel sequencing suits NGS very, very much. Okay. So let's start by looking at the common attributes that most of this, of course, now high throughput sequencing, you have different platforms. Okay, the main ones that comes to mind will be your you have Roche 454, you have your MiSeq, Illumina MiSeq high seq. You also have Ion Torrent, you have your PacBio. So all these are different platforms that allow high throughput sequencing. Okay, but you will learn also that all of these methods have their pros and cons. So depending on what you require, you have to choose the right platform for your work. For example, usually when they compare platforms, they will compare in terms of length of reads. So how long? Okay, so let's so they'll compare you can do the Roche 454. You have your Illumina MiSeq, okay, and high seq. You have your Ion Torrent. You have your PacBio. Okay. Even now you and etc. Lots of different platforms. So when they compare the different platforms, they will look at the read length. and then they will look at your error rates, okay, and how good they are at handling certain sequences, okay. So when you look at read lengths, so how long can these platforms generate? How long is the read that these platforms can generate? For example, for Roche, it can go up to 1000 base pair. Illumina MiSeq is very short, 250 base pair. But if you do the reverse sequencing also, you can get 500 base pair. Iron Torrent is also around that. PacBio is very long, up to 1500 base pair. Okay, but then when it comes to error reads, PacBio has among the highest error reads. Roche also. Error rates are very low in MiSeq and HiSeq. Okay, and they also the cost per base. So cost per base also, Roche 454 is very expensive because of the consumables that they take up. Okay, so now MiSeq, HiSeq have probably the best cost per base, most efficient sequencing cost per base pair. Okay, so most of the high throughput sequencing that we do usually use this MiSeq HiSeq platform. Okay, so in UM we actually have this MiSeq, HiSeq, Iron Torrent, and PacBio platforms. We don't have this Roche 454 platform. So they use PacBio mainly for human genome work or full genome work. Okay, because the reads are long, but because of the error, they will usually. So when you use PacBio, okay, nowadays when scientists use PacBio, they use it to read the whole genome of a microorganism or maybe one full chromosome of a human or human genome project. Okay, so even though the error rates might be high, what they do is they run concurrent with a HiSeq. So whatever possible errors from PacBio, they can correct using the HiSeq data. So that's why usually they run these two together. Okay. But for microbial ecology, when we're looking at diversity nowadays, the main platform that we will keep seeing being repeated in the publications and journals are MiSeq platforms. Okay, so all of them share a specific flow of how they process the DNA. Okay, so the first thing is they will share the DNA. Okay, once you have your environmental DNA, there's random fragmentation of starting DNA. So they will share the DNA. They need to share it to suitable lengths because if it's too long, it will not work in these platforms. So they need to share it to suitable lengths and then start the library construction. Library construction, so they will use linkers and adapters so that they can recognize the different parts of the sample. Okay, so like for example, because it doesn't make sense to run one sample for a MiSeq run. It doesn't make sense to run one sample because the amount of data possible from one sample, one environmental sample, is only maybe 1 gig. But the whole platform, the whole MiSeq platform can probably generate more than that in one run. So what the companies usually do is they will actually mix the samples together. Mix samples from different places together and then run one shot. But how do they know which sample is which? That's where the adapters come in, the linkers come in. They will have specific barcodes in the gene sequence, in the linkers and adapters, so that they know from the sequencing data later uh, where's this data from and from what sample they are from. Okay, so library construction. Then once they have done library construction, then they start the library amplification. Now it's usually on a solid surface, either bead or glass. So beads usually for Roche 454, glass is for MiSeq HiSeq platforms. So this is also called clonal or bridge amplification. Okay, so once you've done this process, you have polonies. Okay, so they term it as a polony. So one polony will be a group of DNA strands that are the same. Okay, then once you have your bridge amplification done, that's where you do your sequencing reaction. So this is the reaction is by sequencing by synthesis. So this is different from your Sanger sequencing. So what actually happens is when you sequence by synthesis, you add your adenosine, guanine, cytosine, and thymine. Okay, and each of these nucleotides will have a different color. So when it's incorporated, a different color will fluoresce. That's why you need a very good imaging system where they are able to detect the different colors that is being released. Okay, so that's how they are able to carry out the sequencing by synthesis part. Okay. Once you've obtained the sequences from each polony, you have lots of reads now, easily 60,000 reads for one sample. If you're looking for a genome, then you have to start contigging, which means you have to scaffold them. You have to lay them where the sequences overlap. You have to scaffold them so that you can start building the genome of the organism. So you get hundreds of thousands to hundreds of millions of reaction image per instrument run. So that's why it's called massively parallel sequencing. The sequencing occurs in a parallel fashion together. Okay. Okay. So now let's look at the. So this is the. So this is a comparison between Sanger sequencing and the NGS sequencing. So initially it's fragmentation into a suitable length, and then here you ligation and linking and using adapters and linkers. Okay, you're creating your library. Okay? here the library is connected like this, and then generation of a polyny array. However, in Sanger sequencing, your library is created via cloning and amplification. Okay, so each sequence, each DNA fragment that is different is inserted into these different colonies. Okay, that's why they use the word polyny here to reflect what you're seeing here. So here, each strand is possibly different, and then they'll attach this to either a glass bead or a glass platform, and you have clonal amplification here, and each strand will multiply, will into a polyny. Okay, then cycle sequencing for Sanger, where you terminate the sequencing reaction using the dideoxynucleotides, okay, of different colors, and then from the colors you can see the sequence of the DNA that you the DNA fragment that you have. However, for NGS, all these are linked onto a glass plate. Okay, so each dot that you see now is a polyny. You have to create a polyny. You have to amplify it to become a polyny, so that whatever incorporation of color, incorporation of a nucleotide that occurs, you are able to see the color clearly. Okay, the camera are able to capture. So every cycle will see a different strand. So here you only see one sequence from one DNA. Okay, but here this is massively parallel sequencing. All this sequencing occurring at the same time. Okay, so that's the power of the imaging system here, and the power of the software that is able to handle the amount of information that is generated here. So you can see clearly that the amount of the cost per base pair has dropped tremendously. So ABI 3730 is still the one that utilizes capillary Sanger sequencing. So even after Roche 454, which is supposedly an NGS, next generation sequencer, the price still didn't drop that much because the price of the cost of the consumables are very high. Okay, but once the high-seq MiSeq system came up, the price went down significantly. Okay, not only the price that came down, but the speed where you can get your results. Okay, nowadays you can get a full genome, full human genome within just no, you can get a full microbial genome within just a few days. Okay, so not only has the price came down, but the amount of output per day, see, the amount of output per day has increased tremendously. Okay, so that is the present state of sequencing now. Okay, so very a lot of data, enormous amount of data, easily can overwhelm a lot of computing systems. But then the problem with so much data is, with all this data, there are error rates. So there's also possible rubbish that is being produced alongside all those sequences. So as a scientist, you have to be very careful. You have to be able to separate out what is data and what is not, what can be used and what it cannot be used. Okay, any questions?
No questions so far, Doctor.
Okay, so now we start a bit of detail into Pyro Sequencing, Roche 454 system platform. So this is the one that started it all, primarily because uh, their platform followed a different paradigm. Okay, so before 454, Roche 454, most of the sequencing platforms follow the Sanger dideoxy termination method. So what Pyro Sequencing does is, it was the first platform to implement sequencing by synthesis. Okay, so now we will look into in detail how this works. Okay, sequencing by synthesis. So you will have your single-stranded template, and then your sequencing primer will be hybridized to it. Okay, then you see here that there are enzymes to be used: polymerase, ATP sulfurylase, luciferase, and epirase. Okay. You also have substrates, your adenosine 5-phosphosulfate and luciferin. Okay, so what happens first is, you have your polymerase here, then you add your nucleotide, DNTP, deoxynucleophosphate triphosphate, to the reaction. So if the sequence is, let's just say the sequence is so the complementary sequence here will be your cytosine, okay, CTP. So if you add CTP to it, it will incorporate by the polymerase. Okay, the polymerase will catalyze the incorporation of the complementary DNTP. So when it incorporates, it will release a pyrophosphate. Okay, remember the CTP is triphosphate. Okay, CTP is triphosphate. So when you incorporate the cytosine into the DNA fragment, you will release a pyrophosphate. Okay, 2 P's here. Okay, then the pyrophosphate that is released will react with adenosine 5-phosphosulfate and will be catalyzed by sulfurylase, the enzyme, into ATP. So you know ATP is an energy currency. And what this ATP does is, it will activate the luciferase enzyme to generate light. Okay? And it is this light that is captured by the CCD chip, by the imaging chip. And so the height of each peak is proportional to the number of nucleotides incorporated. And it is the light emitted that is detected by the imaging system. So this is important. So each incorporation event will release pyrophosphate in a quantity equimolar to the amount of incorporated nucleotide. Okay, what does this mean? This means that if this represents one cytosine that is being incorporated, if the height here is double, that means 2 Cs are incorporated. If the height here is triple, that means 3 Cs are being incorporated. Okay, so that is important. So the height of each peak is proportional to the number of nucleotides incorporated. Okay, so once the light is detected, apirase nucleotide degrading enzyme will be added to degrade all the unused or unincorporated or nucleotides. Okay, then when the degradation is complete, another nucleotide is added. So what you have here is sequencing by synthesis. Any questions?
No, doctor.
Okay. So how does it work? So the disadvantage of a Roche 454 system is you can only add one nucleotide at a time. So you add one nucleotide with all the enzymes, it's incorporated, then apirase will degrade everything. You have to flush out the system and add a new one, C. Okay? If C is incorporated, add apirase, degrade everything, remove all the enzymes, and add T. Okay? T. There's no T, it's not incorporated, but still, you have to add apirase here, degrade everything, and add the next one. So this cycle continues. Okay, but when you have two incorporation, you will see the peak is double. So because it's in equimolar reaction. So if the peak here is C is double, then you know it's a equimolar. So from here, from the light detected, you can determine the sequence of your DNA. So it becomes GC, A, GG, CC, T. Okay, from this sequence. From this sequence of lights, light emitted. So this is where the weakness of pyro sequencing also occurs, because it uses a lot of enzymes, it uses a lot of substrates. So even when for every run you have to degrade all the enzymes, all the nucleotides that you added. Okay, that's why the cost of pyro sequencing is relatively high compared to the other platforms that we have now. Okay. Also, another problem is it doesn't work well with sequence of with poly A or poly G or poly C reads. Okay, so if you have a lot of G here, let's just say you have 5 continuous G nucleotides, you will incorporate some error here because there is a range of detection where is no longer where the imaging system can no longer detect. Okay, so the light emitted no longer correlates with the amount of nucleotide incorporated. Okay, so pyro sequencing doesn't do so well with long nucleotides of the same reads or the same nucleotides. Okay, so it also doesn't work well. It also uses a lot of consumables. Okay, so this is how the pyro sequencing is being incorporated in the NGS platforms. So for MiSeq, they use a glass platform, but for pyro sequencing, they use beads. Okay, they use beads in order to incorporate the same polyny. So every bead will have the same DNA fragment. Okay, then when you synthesize it, then when the synthesize, then every bead will be placed into a single vessel. So during the synthesis, the light from each well will be captured and will be incorporated into the software system. Okay, so you can get up to 400,000 reads in parallel. Okay, 400,000 reads. Okay, so this is another picture that shows how the 454 works. Okay, any other questions?
No question from me, Doctor.
No question, okay.
Okay, so next we go for the Illumina platform. Okay, previously known as Solexa. Okay, I think it was bought up by Illumina. So here the polyny, the clonal amplification, bridge amplification allows the the same DNA sequence to group together at one spot. Okay, so each will be grouped together so that when you're carrying out the sequencing by synthesis, you can see the color clearly. Okay, so DNA fragmentation and then library preparation by adding adapters and linkers. Okay. So once you have your sequencing ready library, you have to run through the flow cell, a glass type of platform, okay, where the adapters will attach to the flow cell. So this is where the clonal or bridge amplification occurs. Okay, so through several steps, sequencing will occur to generate a lot of clonal copies. Okay, either forward and reverse, all come together at one spot. So this is another figure that shows the same thing. So once you have done your library preparation, you run through the flow cell and the DNA will attach to the flow cell. And once the attachment has been carried out, you start doing the bridge amplification, bridge or clonal amplification. And once they have amplified, you can see that this all DNA will have the same sequence, each at one spot. Okay. Then when you're carrying out the sequencing by synthesis, here the nucleotides are linked to a fluorochrome. So when there's incorporation, the light will fluoresce. But the good thing about this is all the nucleotides can be added. You don't have to sequentially add them. Okay. It is what that is incorporated that will fluoresce. So you don't really have to add only one and then flush, add only one and then flush like the 454. Here it's more efficient. You add all four at once, and whatever that is being incorporated, you will see the fluorescence of the light, different color for different nucleotide. Then from there, the computer and the imaging system will remember the spot of the light, and they will detect the different color changes. Then from there, they can actually determine the sequence for each polony. Okay. So for high seed, you can see the different runs and the amount of data being produced easily in hundreds of gigs. Okay. So this is a single molecule sequencing. Single molecule real-time sequencing. This is the one that is being used by PacBio. Okay, but you can read this if you want. Okay, but initially is the DNA polymerase is linked to the platform. Okay, the detector is at the bottom, and when there's incorporation, the detector will detect the light change. So it uses a kind of a sequencing chip, not a flow cell. Okay, so although the reads are extraordinary long reads, okay, but the accuracy is still not very high. Okay. Oh, this is